Hello everyone, welcome to the discussion of your molecular biology and diagnostics. For today's lecture, we will be discussing your Module 5 up until the Module 9. So again, ang discussion po natin will be from Module 5 up until the Module 9, but please do not worry as this discussion will be divided into at least 3 to 4 parts. Plus meron tayong mga i-skip na PowerPoint slides kasi mga good to know lang sila. And then, pwede natin silang gamitin for reviewer na lang. So, yung discussion lang natin will be concise. Okay? So, kung ano yung mga importante, and then yung mga medyo feeling ko maguguluhan kayo or hindi magiging clear yung topic sa inyo, yun yung i-discuss natin. Pero dadaanan natin sila module by module from 5 up until the module 9. Okay? So, for our Module 1 up until the Module 4, you were able to discuss the DNA as well as the introduction to your molecular biology. So, nag-focus tayo sa replication, the DNA structure, DNA replication, the introduction to your molecular biology, and other tests to diagnose different diseases involved in the molecular biology. But for your Module 5, magpo-focus tayo sa RNA. Okay, basically the RNA structure. Also, for Module 1, we were able to discuss the central dogma of life. So, kung kabisado mo yun, and then sinapuso mo si central dogma of life, pwede mo siyang gamiting concept, padadala mo siya, and then makakatulong yung principles na naaral mo sa central dogma of life to further enhance your knowledge in central dogma of life for the upcoming topics which will be discussed later on for the Module 6 up until the Module 9. But for our Module 5, focus muna tayo sa RNA structure. Okay? So your RNA is also a nucleic acid. Ano pa ba yung mga examples ng nucleic acid natin? Yes, tama po yung nasa isip nyo, DNA. So DNA and RNA, they are basically nucleic acid and they are being comprised of nucleotides. So ano ba yung mga primary components of each nucleotides? Wag natin kalilimutan si phosphate group, si sugar group, your pentose sugars, and then the nitrogenous bases. So tatlo lang naman yung tatandaan mo. Meron kang phosphate group, meron kang sugar, and then the nitrogenous bases. So basically, same lang yung structure, yung component ni DNA to RNA, meron lang silang slight differences. Sa phosphate group, wala naman po masyadong kinaiba. Sa sugar, ano po yung pinaka-main difference nila doon sa ating single atom, which is the oxygen? Hindi pa po. Sa ating pentose sugar. Si DNA, deoxyribose, wala siyang oxygen doon sa ating pangatlo, sa pangatlong pentose sugar, di ba? 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Sa pangalawa pala, sorry. 2, 3, 4, and then the 5. Yun yung main difference nila. Meanwhile, for the nitrogenous bases, ang pinagkaiba naman nila is yung ating uracil. Si RNA may uracil, sa ating DNA, ang counterpart niya ay si thymine. Next. And then your RNA has different sugar and then the uracil instead of thymine. So yun yung slight differences of your RNA and then your DNA. And then please do not forget that your RNA is single-stranded compared to your DNA, which is double-stranded. And then the RNA molecules have extensive base pairing, which produces secondary and tertiary structures that are important for RNA function. And then the adenine with uridine, it requires 2 hydrogen bonds. And then the guanine with cytosine, it requires 3 hydrogen bonds. So ito po adenine and then uracil yan, sorry. So kung matatandaan for the DNA, ano ba yung base pairing doon that requires 2 hydrogen bonds only? That will be your adenine and then the thymine. So wala naman nag-iba, si guanine and cytosine for the nitrogenous bases, 3 hydrogen bonds pa rin po siya. And then the messenger RNA or what we call the mRNA, it contains a cap structure and then the poly-A tail. And then the cap consists of methylated guanine triphosphate or what we call the GTP attached to the hydroxyl group on the ribose at the 5' end of the mRNA. And then the N7 is the guanine. In the guanine is methylated and then the 20 hydroxyl groups at the first, second ribose of the mRNA also may be methylated. Next, your rRNA, it contains many loops and extensive base pairing. So kung natatandaan sa inyong DNA, meron po silang base pairing. As for your RNA, sa rRNA, meron din po tayong tinatawag na base pairing. And then the rRNA molecules differ in their sedimentation coefficient, yung letter S. So kung mapapansin nyo sa ating mga pictures later on, meron ka makikita na number, tas may letter S. Kunwari, 23S. Okay, so wag malilito, ang ibig sabihin po nun, yun yung sedimentation coefficient. At para saan po yun? They associate with proteins to form your ribosomes. And then the prokaryotes have 3 types of rRNA. Please do not forget these 3, your 16S, the 23S, and then the 5S. That will be your 3 types for the prokaryotes. Meanwhile, for the eukaryotes naman, meron kang 4 types of cytosolic rRNA. Ang mga tawag naman doon, sina 18S, 28S, 5S, and then the 5.8S. Okay? And then the mitochondrial ribosomes are similar to prokaryotic ribosomes. And then lastly, the tRNA has a cloverleaf structure and contains modified nucleotides. tRNA molecules are relatively small, containing about 18 nucleotides. And then ito po yung sinasabi ni ma'am na may mga sedimentation coefficient. Ito po yung para sa ating prokaryotes and then the eukaryotes. They are involved in the synthesis of the ribosome. So kung may mga mapapansin kayo mga proteins po dito sa ating diagram. And then ito, mga nice to know lang to with regards to the structure of the RNA. We will not go into detail of the cloverleaf structure and then the D-loop, the anticodon. Ang mahalaga, alam niyo lang po yung structure of the RNA. Okay? And the next, we will now move forward to your next module, which will be about your transcription. So kung matatandaan sa ating central dogma of life, ano po ba ang ating transcription? So according to the central dogma of life, the flow of genetic information in cells is almost exclusively one way. So from the DNA, pero meron tayong pwede maganap na replication dito. So from DNA to DNA, replication yon. So kapag ka naman ita-transcribe mo na siya from DNA to RNA, anong tawag na doon? Transcription. So kung from RNA, ita-translate mo na siya papunta sa proteins, translation na po yung tawag doon. Diba? So recall lang natin yung principle behind the central dogma of life. And then, sabi nga daw dito, yung flow ng genetic information in cells is almost exclusively in one way. So lahat ng mga living things or non-living things na merong DNA, they basically go the same way or exclusively in one way, which involves the central dogma from the DNA papunta sa inyong mga proteins. And then for the transcription, DNA-directed RNA polymerase, mamaya may encounter natin to, puro RNA polymerase yung mababasa natin. And then sa translation, involves si ribosomes. Again, ribosome is a part of your cell. And then what does our ribosomes do? Your ribosomes, again, pakirecall po ang ginagawa ng ribosomes kung nasaan sila, nasa organelles. Diba mga organelles yung mga yan na naka-embed sa ating cytoplasm. And then your ribosome, involved siya sa protein synthesis. Kaya nabanggit po dito si ribosomes inside the cell. Okay po. And then the synthesis of RNA or what we call the transcription. So ito yung mga processes involved in the synthesis of the RNA or what we call the transcription. So bago siya maging RNA, manggagaling muna siya dun sa DNA, di ba? So DNA, tapos magiging RNA, tawag doon transcription. So dito na pumapasok si RNA polymerase. RNA polymerase can initiate the synthesis of new chains. A primer is not required. Again, let us just read that statement. RNA polymerase can initiate the synthesis of new chains. A primer is not required. So if you can recall from the DNA replication, bago tayo mag-initiate ng ating pagbukas ng replication fork, diba kung matatandaan, ano po yung kailangan doon? para mag-initiate yung pagbukas ng bubble fork, ng ating replication fork, bago yung another template, makagawa ng mother and then ng baby, bago sa buong process ng replication. If you can recall, ano po yung kailangan para mag-initiate yung pagbukas ng ating replication fork? Ano po yun? Yun, tama po yung inyong mga nasa isip. Yung single-stranded binding protein, yung SSBP, yun yung mag-i-initiate ng pagbukas ng replication fork. Pero yung pinaka-enzyme na involved para magbukas yung replication fork, yung dalawang strand para magbukas siya, yun yung mga tinatawag nating helicase. Okay, recall lang. But for our RNA, meron tayong tinatawag na RNA polymerase that can initiate the synthesis of new chains, but it does not require a primer. Next, the DNA template is copied in the 3' to 5' direction. Sorry, hindi to 30 at 50. 3' to 5' direction, and the RNA chain grows in the 5' to 3' direction. So kung makikita nyo, parang similar lang siya with our DNA replication. Meron lang mga naibang proteins, meron lang mga naibang enzymes involved, but basically parang same lang sila ng kanilang process. And then the ribonucleoside triphosphate, the GTP, ATP, and then the triphosphate and the cytidine triphosphate serve as the precursors for the RNA chain. And then the process is similar for your DNA synthesis. And then the synthesis of RNA in the bacteria, magkaiba po sila for the prokaryotes and then the eukaryotes. So, paki-take note na lang yung mga differences in the synthesis of RNA when we're talking about the prokaryotes and then the eukaryotes. So, for the synthesis of RNA in bacteria, the RNA polymerase of E. coli contains 4 subunits. Pakitandaan po sila. Ito po yung mga yon. Again, pakitandaan po yung mga mahalagang enzyme involved in the synthesis of RNA bacteria. Meron tayong tinatawag na core enzyme. Core enzyme. Meron tayong pumapaloob dyan na dalawang alpha. Tama ba? Alpha units. Meron tayong beta, dalawang alpha, dalawang beta, and then meron tayong tinatawag na omega unit. Okay, so for the core enzyme, hindi lang maintindihan yung sulat ni ma'am, pero meron po tayong tinatawag dito na dalawang alpha, dalawang beta, and then omega unit. And then meron din tayong tinatawag na sigma subunit. Okay, pakitandaan po yung mga sinabi ni ma'am na yun, which are all required for the initiation of the RNA synthesis like what is indicated in our PowerPoint slide. So next. Ito na yung sinasabi natin parang kanina lang. Meron lang tayong picture dito, illustration on how the RNA polymerase works. Okay, so meron po tayong ganito. In the RNA polymerase, it is the main enzyme involved in the transcription. Pakitandaan, wag na wag kalilimutan. Ano yung main enzyme involved in the transcription? It is what we call the RNA polymerase. It reads one of the DNA strands and adds complementary nucleotide bases to make an mRNA transcript. And then the mRNA will later be translated into protein. So mahalaga po si mRNA or what we call the messenger RNA for the DNA to be translated or to be transcribed into RNA, which will later on be translated into a protein. So, mahalaga po si protein sa structure ng ating katawan kasi basically madami siyang functions. So, from the DNA, mahalaga yung mga nucleotides natin, yung mga nucleic acid natin, yung ating mga DNA to make or to synthesize a protein. Kasi nga, sobrang daming function ni protein sa ating katawan. So, basically, we are being comprised of a lot and a lot of proteins. So after that, the information constituting an organism's genotype is carried in its sequence of bases, and then the DNA is transcribed into RNA, which is translated into a protein. So, kung babalikan lang ulit natin yung principle ng central dogma of life, it basically tackles that the DNA will be transcribed into RNA and then the RNA will soon be translated into a protein. So, if you will look closely, ito yung itsura ng ating RNA transcripts of DNA microscopically. And then, if you can recall, ang ating DNA replication, it involves 3 steps or 3 major processes, naming our initiation, elongation, and then determination. So, for the transcription of the DNA to RNA, basically same lang sila ng process. Yung tatlong nabanggit natin na 3 major processes, involved din siya dito sa ating major transcription. So let us just read the step description and then the characteristic. Number 1, formation of a close promoter complex. The characteristic is that number 1, RNA polymerase binds to the promoter and forms a close complex. And then the sigma subunit directs polymerase to the promoter. And then the conversion of the closed promoter complex to an open promoter complex. Then the beta and sigma subunits initiate separation of DNA strands 10 to 17 base pairs. So kung matatandaan, meron tayong core enzymes na tinatawag for the prokaryotic cell transcription. So pakitandaan yung primary components na yon. Meron tayong 2 alpha unit, 2 beta at 1 omega. Meron din tayong tinatawag na sigma subunit factor. Next, the polymerization of the first few nucleotides, it involves these characteristics. And lastly, the promoter clearance, it involves the RNA to DNA stable hybrid complex, contributes to the stability of the elongation complex. So kung matatandaan, tandaan po ito, meron tayong tinatawag na promoter region. So for the DNA replication, meron tayong tinatawag na SSBP, single-stranded binding protein. Yun yung pinaka nagsa-start na protein para magkaroon ka ng initiation process para makabuo ng replication fork at maghiwalay na yung 2 template strand. For the transcription of a prokaryotic cell po ito, meron tayong tinatawag na promoter region. And then ito yung itsura niya. Ito yung tinatawag natin na core enzyme. And then ito yung promoter search. The holoenzyme continues to rebind loosely into the DNA in search for the promoter. And then itong enzyme na to, kakabit siya dun sa inyong template. And then close promoter complex formation. It occurs once holoenzyme has found and bind to a promoter sequence. And then open promoter complex formation naman yung tawag pagka it binds between enzyme and DNA titans, melting a region of DNA to form an opening. Next naman, kung tapos tayo sa initiation, meron tayong tinatawag na elongation, yung pangalawang major process. Elongation is the subsequent binding of ribonucleotides to the growing RNA chain. So yung RNA chain natin, it will be read from 5 to 3. And then the RNA polymerase and transcription bubble direction will be from the 3' end to the 5'. And these are the step descriptions and then their characteristics. Meron tayong tinatawag na dissociation of sigma subunit, core enzyme elongates mRNA, and then the RNA polymerase maintains short melted region. So kung matatandaan, meron din tayong tinatawag na dito na topoisomerases. What does your topoisomerases do? Sila yung parang nag-unwind para mapigilan yung supercoiling ng ating mga DNA templates or DNA strands. For the elongation of RNA, applicable pa rin po ang ating mga topoisomerases to prevent the supercoiling. Okay? Coiled siya, pero coil ang ating mga DNA and RNA, but supercoiling is bad for that specific template. So, dito yung ating topoisomerases para mapigilan yun. And then these are the characteristics of elongation. It is non-uniform and unsteady. Again, the characteristics of your elongation is non-uniform and unsteady. The RNA polymerase movements in the DNA template involves pausing and then the backtracking. So let us just read the description of pausing. It allows spacing of the translation. And then it is very important in the attenuation and aborting transcription. And then it is important also in the transcription termination. Ano po ba yung attenuation? Attenuation, it regulates expression by causing premature termination of transcription of the operon when the operon's products are abundant. Next naman po is the backtracking. It is for proofreading. It extrudes 3' end of RNA out of the polymerase and then removed by nuclease activity of the polymerase. Yung proofreading, yun yung binabalikan natin yung mga nabuo nating mga base pairs to check pag merong kakaiba. Okay, so meron dong different na hindi dapat siya yung ka-base pairs niya. Yun yung ginagawa natin sa proofreading way back sa ating DNA replication. So for the elongation of the RNA, start siya ng transcribing. Okay, yung transcription natin. And then the pausing, and then the backtracking, kung saan involved na yung ating proofreading. And then last na major step natin, so tapos na tayo sa initiation and elongation, nandito na papasok yung termination ng transcription. Under po ito ng transcription. Termination involves specific sequences downstream of the actual gene for the RNA to be transcribed. Meron tayong tinatawag na intrinsic termination and then the RHO-dependent termination.